This video will cover subtopic 2.7, transcription and translation. Here is a list of understandings, applications, and skills that will be covered related to transcription and translation. So in this process of transcription and translation, it's basically taking DNA, using that DNA to build an RNA molecule, and that RNA molecule is used to create a protein. So basically we're going to look at how we go from gene to protein. Now the path from gene to protein has 3 steps in it. In transcription, molecules of RNA are going to be produced from the DNA in the nucleus. So DNA is used to make that RNA. During RNA processing, that RNA is modified before leaving the nucleus, and non-protein coding regions of the RNA strand are removed. So there'll be some modifications to that RNA before it gets used to make a protein. And then in the last step, in translation, the RNA molecules are used as a code for protein assembly at the ribosome. So that RNA encodes that message of what amino acids need to be added in what order. So what is transcription? So transcription is a process by which an RNA sequence is produced from a DNA template. Where does transcription happen? In prokaryotes, it happens directly in the cytoplasm. In eukaryotes, it will happen in the nucleus, or it can happen in mitochondria or chloroplast. But for our purposes, this will be directly related to transcription in the nucleus. So the steps in transcription are that there's an initiation step, an elongation step, and a termination step. So 3 steps in creating an RNA molecule. In the first part, in initiation, in the nucleus, we'll have RNA polymerase, which is an enzyme that has polymerase activity as well as helicase activity. So it can both unwind the DNA and create the RNA molecule. Will attach to the DNA sequence at a location referred to as the promoter. So there is a region of DNA that will allow the RNA polymerase to attach. It will tell the RNA polymerase this is a gene that needs to be transcribed. Once it is attached, then the elongation stage starts. So this is where RNA polymerase will synthesize the RNA transcript. So once the RNA polymerase is attached, it will start moving down the DNA strand, creating an RNA transcript. So the red line here is that RNA molecule that is being created during that elongation step. Having a closer look at this, only one region of DNA strand is used as a template. So as the DNA is unwound, the RNA polymerase will use the template strand of DNA. So there's a coding strand, and then a template strand, and it's the template strand that gets used to build the RNA molecule. So the RNA polymerase will take RNA nucleotides, and remember these are nucleotides that will have a hydroxyl group on carbon 2 in that molecule. Those RNA nucleotides then get incorporated or used by RNA polymerase to create that new RNA strand or that RNA transcript. And so RNA polymerase will read the sequence on the template strand to tell it what RNA nucleotides need to be incorporated. Remember that in RNA, we find the nucleotide uracil instead of thymine. So wherever there is an A U will be incorporated. And wherever there is a T on the DNA, an A will be incorporated. So on the RNA, instead of T, you have a U. In the final step, the termination step, this is where the RNA polymerase will reach the transcription terminator signal on the DNA. So there is a signal at some point on that DNA that says, that tells the RNA polymerase to stop making an RNA transcript. When the RNA polymerase reaches this terminator signal, it will then dislodge the growing RNA strand. So the RNA strand will be released from the RNA polymerase, and the RNA polymerase will fall off of that DNA sequence. So here we have our completed RNA transcript that is now ready to be modified and then exported from the nucleus. So once transcription is complete, there is some RNA processing that goes on. So there is modification to the RNA sequence before it is then exported from the nucleus into the cytoplasm, where translation can then happen. So this is where we find our ribosomes out in the cytoplasm, and thus where translation occurs. Before we talk about the process of translation, we need to talk about the three types of RNA that can be made. So the first type is messenger RNA, or mRNA, and this is the RNA that we assume has been made, or the RNA that is being made through transcription. So it is basically a transcript copy of the gene used to encode a polypeptide. So that RNA, or that mRNA sequence, will contain the code for a protein. Another type of RNA is referred to as transfer RNA, or tRNA. This is a cloverleaf-shaped sequence of RNA that will also carry an amino acid at its end. So here we have our tRNA. The green part is the RNA molecule that bends and twists and forms like this cloverleaf shape. And then at the end is an amino acid attached onto it. On the transfer RNA, there is a region of interest here referred to as the anticodon sequence. It is that anticodon sequence that will help build to the polypeptide. The third type of RNA is referred to as ribosomal RNA, or rRNA, and is found within ribosomes. So as I mentioned before, transcription will occur in the nucleus where the DNA is located, and then once the RNA has been made, that mRNA moves to the cytoplasm where translation can occur. So with translation now, this is the synthesis of polypeptides or protein on the ribosome. So just a reminder of what a ribosome is and the different parts of that ribosome. So a ribosome is composed of two halves. There is a large subunit and a small subunit. During translation, the ribosomal subunits will assemble together. So those two parts will come together on the strand of messenger RNA. So messenger RNA plus those two subunits must assemble together before translation can begin. So the small subunit will bind to the mRNA, and then the large subunit has binding sites for transfer RNAs, and will also catalyze the peptide bonds between amino acids. So the amino acid sequence of the polypeptide is going to be determined by the mRNA according to the genetic code, and we'll talk about that genetic code on the next slide. So the length of the mRNA molecule will vary, and because of that, the length of the polypeptides will vary. The average length for mammals is approximately 2,000 nucleotides, which codes for about 730 amino acids. And remember that only certain genes in a genome need to be expressed. So this is based on cell specialization and the environment of that organism or that cell is located in. Cells that need to make large amounts of a particular polypeptide or protein will make many copies of the mRNA for that polypeptide. So, as an example, beta cells that are found in the pancreas will make lots of copies of the mRNA transcript for that protein, insulin, because it's making lots of insulin. So, the way that mRNA translates into a protein code is through the use of what's referred to as codons. And those codons are what make up the genetic code. So the genetic code is basically a set of rules by which information encoded in the mRNA sequence is converted into proteins. Essentially, when we say converted into proteins, what is that amino acid sequence of that protein? So codons are essentially what will code for those amino acids. So codons are triplets of bases which encode a particular amino acid. When we say triplets of base, we mean 3 bases in a sequence. So for example, GAG, that triplets of base will encode for a particular amino acid. Or if you have UUA, that will encode for a different amino acid. So based on the fact that we can have G, A, C, and U as our letters in any arrangement of triplets, this will give rise to a total of 64 different codon combinations. Now, even though there are 64 different codons, those codons can translate for 20 amino acids. So there's more codon combinations than there are amino acids possible. So what this means is that different codons can translate for the same amino acid. So in this regard, the genetic code is said to be degenerate, or there are redundancies in those codons. So this allows for any errors to occur in the mRNA sequence, but not necessarily affect the amino acids that get incorporated. So now the order of the codons that are in an mRNA sequence will determine the amino acid sequence for a protein. So looking at all these different combinations of codons in the chart that's on the slide, gives you the different combinations of codons and then which amino acid corresponds to that codon. So a coding region will always start with a start codon, AUG. So if you find A, U, and then G, that start codon is always methionine, and methionine is thus the first amino acid in all polypeptides. Coding regions always terminate with a stop codon. So stop codons do not code for an amino acid. So it's basically like a blank tRNA, no amino acid attached. And that blank tRNA, or that empty tRNA, will cause the release of the polypeptide from the ribosome. Will basically tell the ribosome to stop making a protein from that mRNA sequence. So how translation happens is that those ribosomes will act as the binding site for mRNA and tRNA. Remember, tRNA is that clover leaf RNA molecule that also has an amino acid attached to it. So there will be a tRNA for each different amino acid. And what the ribosome will use is that electrobinding site for the mRNA and tRNA, and will catalyze the formation of peptide bonds on the polypeptide. So the tRNA molecules have what's referred to as an anticodon sequence on them of 3 bases. that anticodon sequence is going to be able to bind to the complementary codon of the mRNA. So in the diagram here, the blue segment is going to be our ribosome. We have our mRNA passing through the ribosome. What the ribosome will do is that once it encounters that start codon, it will start then reading triplet bases. So it'll read a set of 3 bases on that mRNA. And it will then allow in a transfer RNA, in this case CUC, that is complementary to that messenger sequence or that codon sequence on the mRNA. So the tRNA carries a codon sequence, that anticodon sequence that is complementary to that codon sequence on mRNA. And then the mRNA, or the tRNA, sorry, is going to be carrying an amino acid attached to it. So once that transfer RNA enters into the ribosome, it'll be adjacent to another amino acid, and then the ribosome will catalyze the formation of a peptide bond between those 2 amino acids. And so the ribosome will continue moving down the mRNA, allowing transfer RNAs in, building that chain of amino acids. So those tRNA molecules, as I mentioned, do carry the amino acid corresponding to their codon, and the mRNA has the sequence of codons that specifies the amino acid sequence of the polypeptide. The main events of translation that have to occur are that first, the mRNA has to bind to the small subunit of the ribosome. The complementary tRNA to the first codon, and remember AUG is that start codon, will then bind on the mRNA to the ribosome. So here we have our first tRNA that is complementary to that start codon, and that tRNA contains that amino acid methionine. A second transfer RNA that is complementary to the second codon of the mRNA binds. So here our next codon would be, in this case, GGA. The tRNA that is complementary to that codon will now bind. And at this point as well, the large ribosomal subunit will now attach. And then the ribosome will then transfer the first amino acid to the second amino acid by making a peptide bond. So a peptide bond will be catalyzed between the thymine, in this case glycine. The ribosome will then move along the mRNA. The first tRNA gets released, so it's a spent tRNA, so it gets released or discharged, and then now the second tRNA will now become the first tRNA. Another tRNA will bind with an anticodon complementary to the next codon on the mRNA. So the next codon in the sequence now allows the next tRNA into the ribosome that carries the amino acid coded for by that codon. The ribosome then transfers that chain of amino acids carried by that first tRNA to the amino acid on the next tRNA by making a new peptide bond. So a new peptide bond gets formed. Steps 4 through 6 basically get repeated until a stop codon is reached. So the ribosome keeps moving down that mRNA, incorporating transfer RNAs letting transfer RNAs into that large ribosomal subunit, catalyzing peptide bonds until a stop codon is reached. So one of the skills that you'd be expected to be able to do is to be able to use the genetic code to deduce which codons correspond to which amino acid. And so when you're using that code, just realize that the 3 letters are used to indicate each amino acid. So, for example, UCU would code for the amino acid serine. So you have to use a table of mRNA codons, which would be provided for you. You don't need to memorize. And then you would use their corresponding amino acids to deduce the sequence of amino acids by a short mRNA strand of known base sequences. So the first 3 bases in the mRNA sequence for the first amino acid, then the next 3 bases for the codon in the amino acid would be read, and then so on and so on. So if you had the short sequence of CGU, those first 3 codons would be used to figure out what amino acid would be coded for, which in this case would be arginine, according to the chart. And the next 3 amino acids would be used to figure out what amino acid would be coded for, which in this case is isoleucine. Now, if you have to deduce the DNA base sequence for the mRNA transcript, basically the DNA sequence is going to be complementary to the mRNA sequence. So this this RNA sequence would have the complementary DNA sequence of the following letters. Now, because the genetic code is universal, meaning it is used by all living organisms, this allows genes to be transferred between species. And where this gets utilized in science and by scientists, for example, is in the production of human insulin by bacteria. So what scientists have done is they've taken a human cell, they've isolated that insulin gene, so they've obtained that mRNA that codes for insulin, converted that to DNA. They've taken plasmids out of bacteria, and they can take that insulin gene and insert it into the plasmid, creating what is called a recombinant plasmid. That plasmid can then be transferred back into the bacteria, and the bacteria now will perform transcription and translation of that human protein, creating insulin. So this bacteria can then be grown in culture, and then the insulin can be extracted and purified and can be used for treatment of diabetes or type 1 diabetes, for example. So that is the end of this video for 2.7, transcription and translation.